All right, next up we have the small intestine. The small intestine is going to is going to be a long, twisted tube that can be broken down into three regions: the duodenum, jejunum, and ileum. The primary function of the small intestine is going to be chemical digestion and absorption. Chemical digestion is going to be done with many different digestive enzymes, most of which are going to be coming from the pancreas. Our carbohydrates, proteins, fats, and nucleic acids are going to be broken down into their simplest forms. We're going to be breaking down polymers into monomers at this stage. The next step is going to be absorption. Absorption is where we go get those small molecules, those monomers, into the bloodstream. This is going to be done primarily in the small intestine. As we've already mentioned, there are some molecules that can be absorbed in the stomach, but very, very few. By and large, everything's going to be absorbed in the small intestine, from the small intestine into our bloodstream. Now, for absorption to occur, we need a very large surface area of the small intestine, and the surface area of the small intestine is going to be increased by several different things. 1, a pleated lining, 2, villi, and then 3, microvilli. Each villus is going to have a network of capillaries and a lacteal and lymphatic vessel. The capillaries are going to carry away absorbed products of protein and carbohydrate digestion, as well as ions and water. Lacteal will carry away absorbed products of fat digestion in the form of chylomicrons. Chylomicrons are just protein-coated products of fat digestion that are soluble in water. Because as we have mentioned many times, lipids are going to be nonpolar, which means they're hydrophobic, they're insoluble in water. And so we're going to need specialized ways in which we can absorb those lipids. So here we have polysaccharides. Polysaccharides are going to be broken down into disaccharides, and then finally monosaccharides. Those monosaccharides are small enough they can be absorbed through the microvilli and pass through the cells into the bloodstreams, into the capillaries. Proteins, same thing. They're going to be broken down into long chains of peptides and then finally into amino acids and absorbed into the bloodstream. We are going to need something else, though, to help break down fatty acids. We can use bile salts. Bile salts will attach to fat globules. It'll help emulsify them, break them down into smaller pieces, and then we're going to break down those fatty acids and monoglycerides even farther using micelles. Those micelles are going to add a charge to these nonpolar molecules, make them polar, and help them pass into the lacteal to be passed into the thoracic duct. All right, this table is pretty interesting. This table is a table of the major digestive enzymes and what they're going to be responsible for breaking down. Spend a little bit of time looking here. Spend a little bit of time here. I'm not going to quiz you on all of the specifics, but because of some of the labs that we're going to be working on, I would like you to know amylase. I would like you to know where amylase is found, where it's secreted, what it does. Maybe know a few of the enzymes involved in breaking down different carbohydrates. You should know what pepsin is, know where it's located, know what it does. Review trypsin, chymotrypsin, carboxypeptidase. Again, I want you to know mostly where the enzymes are coming from and what they break down. So really focus more on this region of the table. It will help you to know the names of these proteins, but it won't be necessary. So it'll help you study, help you understand what's going on where, if you know what all of the names are. But again, if you just know, generally speaking, the proteins involved in breaking polypeptides into amino acids or into smaller fragments are going to be secreted by the pancreas and absorbed into or passed into the small intestine. And then also know that carbohydrates are broken down in the small intestine as well. So anyway, review this table. Spend a little bit of time with it. Table 15.3 is also going to be very helpful. I do want you to know this table. You should know the different structures of the digestive system. What do they secrete? What do they break down? What is their role? And the site of action of chemical secretion. So the salivary glands secrete saliva into the mouth. That's where we can break down food. We use that water to moisten the food to aid in swallowing. We use the amylase to help break down carbohydrates into sugars. The pancreas is going to secrete into the small intestine. That's where we're going to digest. We're going to digest carbohydrates, proteins, fats, and nucleic acids. The liver is going to secrete bile into the small intestine to help emulsify fats. The gallbladder is going to store bile and release it into the small intestine. And again, that bile is going to help with the digestion of fats. So spend a little bit of time with this table as well. This would be a great one for turning into flashcards, or if you can learn better from it in table form, go ahead. All right, next up is the large intestine. The large intestine is where materials that have not been absorbed by the small intestine are going to pass into. Now here, we're really not looking for food or nutrient absorption. At this point, we're just going to see that we're absorbing most of the water remaining in the indigestible food residue, and then we're going to store and eliminate the rest as feces. So there's very little monomer absorption at this stage. It's not the primary goal of large intestine. So here we have our colon. We're going to review the colon structure just a little bit. We have three regions. We have the ascending colon going up, the transverse colon going across, and the descending colon coming down. The primary function is going to be absorb water and ions. It's going to contain beneficial bacteria that produce vitamins. Materials left in the large intestine after passing through the colon is what's known as feces. Here are the regions of the large intestine, as we've already mentioned, the three regions of the colon. But then we have the cecum. The cecum is going to be a small pouch at the junction of the small and large intestines. The appendix is going to extend from the cecum. The appendix has no digestive function, but it may play an important role in our immune system. Homeostatic imbalance of this particular system might be appendicitis. Appendicitis is inflammation of the appendix. Blockage of the appendix by stool or food or possibly a tumor can allow bacteria to multiply in this region. Treatment typically includes antibiotics and surgical removal of the appendix. Peristaltic contractions will move material through our large intestines. Feces is going to be pushed into the rectum and initiate the defecation reflex. Feces are going to move into the anal canal, and two sphincters must relax in order for defecation to occur. The internal sphincter relaxes automatically as part of the defecation reflex. Here we go. And then the external sphincter, right down here. This is under voluntary control, for the most part. Under voluntary control, which is going to be a necessary part of eliminating waste. Disorders of the colon may be diarrhea. Diarrhea occurs when material passes through the colon too rapidly and not enough water is going to be absorbed out, leaving a very thin, liquidy fecal substance. Constipation is sort of the opposite. The material passes through the colon too slowly. Too much water is absorbed and makes it very difficult to pass the rest of the fecal matter. So what can you do to treat either one of these? I'm going to leave that to you. Pouches. Pouches may occur in the intestinal wall. These are called diverticula. Diverticulosis is diverticula that does not necessarily cause problems, but diverticulitis is when those diverticula become infected and inflamed. Colorectal cancer begins with a polyp inside of the colon. However, that polyp could turn cancerous. Detection must be done through sigmoidoscopy. This is where we can examine the first third of the colon. Really, we can't go any further. We can't go across the transverse colon. But we can go up through the descending colon, the sigmoidoscopy is where we can, yes, only go through the first third, and then colonoscopy is when we can actually go through and explore the entire colon in search of colorectal cancer. That's our large intestine. We're going to move now into some of the accessory organs that we've already mentioned, but we're going to spend just a little bit of time with them. The pancreas, its primary function is going to be to secrete enzymes to digest most of your nutrients. So things like carbohydrates, proteins, lipids, nucleic acids. The nutrients are then going to be absorbed into the small intestine, into the bloodstream. The enzyme secretions are going to be released into the initial part of the small intestine. So we're going to dump everything into the beginning of the intestine and slowly pass everything through. Hopefully by the time we reach the end of the small intestine, all of the nutrients will be absorbed and we can move into the large intestines. The pancreatic juice contains many digestive enzymes, including amylase, trypsin, and lipase. It also contains bicarbonate ions that are going to help neutralize the acid in chyme. This is going to drain from the pancreas into the pancreatic duct, which fuses with the common bile duct before entering the small intestine. Liver is also going to be a important accessory organ in our digestive system. This is going to be our largest internal organ. Its primary function is going to be to produce bile. This is going to be nutrient-laden blood from capillaries in the villi of the small intestine is going to travel through the hepatic portal vein to the liver. The portal system is going to be the vein that links the two capillary beds. The hepatic portal system contains a hepatic portal vein that's going to link the capillary beds of the villi in the small intestine to the capillary beds in the liver. This is where we are going to be moving this is where we're going to be moving all of our nutrients. There are some digestive activities of the liver. The liver is going to produce bile, which is secreted into the gallbladder and small intestine. It's going to help regulate blood glucose levels. It's going to package lipids with proteins to form lipoproteins, which aid in the transport back to the blood. It's also going to help remove poisonous substances from the bloodstream. So the liver actually has many different functions in both the cardiovascular system, the urinary system, and the digestive system. Once the liver adjusts the composition of blood, the blood is going to be returned into general circulation. It's also going to be a place for the storage of iron and some vitamins. There are some homeostatic imbalances that can be found with the liver. Cirrhosis is a homeostatic imbalance of the liver. This is where we see an accumulation of fat and then scar tissue in the liver. This is usually caused by excessive use of alcohol. Hepatitis is inflammation of the liver. This is commonly caused by one of 6 viruses, A, B, C, D, E, or G. We do not have a hepatitis F. We do have an hepatitis G. The liver is going to stops filtering bilirubin from the blood, resulting in jaundice. It's sort of a yellowish tint to the skin and the whites of the eyes. This is usually an indication that something is horribly wrong with the liver. Next up, we have the gallbladder. The gallbladder is an accessory organ. It's going to be a pear-shaped muscular sac that's going to be responsible for storing, modifying, and secreting bile produced by the liver. There are some disorders that occur with the homeostatic imbalances associated with the gallbladder. For example, gallstones. Gallstones occur when cholesterol and other substances build up around a particle. A gallstone is cholesterol that has precipitated out from bile during storage, and it can cause problems if they block ducts. They're going to have to be surgically removed. Here's a diagram of our liver. The role that it plays, we have our gallbladder and its opening into the small intestine. All right, so we're going to spend a little bit of time here. Our last video will contain a summary video of the entire digestive system.